Triple-point cells are widely used in situations which demand accurate temperature measurement. The Boots Company, which has a vast manufacturing complex in Nottingham, recently purchased a rather special cell. We asked Eric Emerson, Chief Instrument Engineer, why?
Why did we buy a triple-point cell? Why indeed? We certainly didn't set out to buy one. The reasons make quite an interesting story.
Before we follow his story, let's pause for a moment and find out exactly what a triple-point cell is and how it works. We're all familiar with boiling, condensation, melting, freezing, and sublimation, which are all examples of phase changes. Phase changes are physical changes between the solid, liquid, and vapor phases, which all stable substances undergo at particular pressures and temperatures. These pressures and temperatures can be plotted to produce a pressure-temperature phase diagram on which the phase changes appear as lines known as phase boundaries. A substance exists as a solid in this region of the phase diagram, as a liquid in this area, and as a vapor in this region. Phase changes are very useful in thermometry because of the associated heat of fusion or vaporization. It is this so-called latent heat which keeps the temperature of a boiling kettle constant, since, at the boiling point, all the heat goes to convert water into steam. A similar thing happens in ice-water mixtures at the freezing point. A loss of heat will lead only to further freezing, while any heat gained will just melt some of the ice. So, heat gains or losses affect only the rate of boiling or melting, not the temperature. The boiling and freezing points of water and other substances therefore provide suitable steady-state environments in which thermometers can be calibrated, usually at atmospheric pressure. The most significant factors affecting how accurately this can be done are the purity of the substance and the steadiness of the pressure. Impurities change the phase diagram, depressing the freezing point and elevating the boiling point. That's why we add salt to the road or antifreeze to the car radiator. A change in pressure causes a movement along the phase boundary to a new point, hence a new temperature, which explains why it's impossible to make a good cup of tea on the top of Mount Everest. The water boils at too low a temperature. The purity problem is overcome by sealing distilled water, once and for all, in a clean glass vessel. A long, narrow re-entrance tube will hold a thermometer. If the vessel is sealed under vacuum, then the pressure is also fixed. Since all substances evaporate to some extent under vacuum means, in practice, at the vapor pressure. If melting or freezing is now established, then all three phases, solid, liquid, and vapor, coexist. The phase diagram shows how this can only occur at the unique pressure and temperature where the three phase boundaries meet. This is the triple point, and for water it occurs at 0.01 degrees Celsius or 273.16 Kelvin. In general, triple points offer the most precise and reproducible fixed points of temperature. So superior is the triple point of water to the freezing or boiling points that these are no longer the definitive points of temperature. The Kelvin has long since been defined in the International System of Units in terms of the temperature assigned to the triple point of water. To prepare a cell for use, it's first cooled in melting ice for about an hour. On removal from the ice, the central re-entrant tube is dried with cotton wool. It's cooled by tipping in small quantities of powdered solid carbon dioxide. That's dry ice at minus 80 degrees Celsius, making sure that it reaches to the bottom of the well and replenishing it as necessary so that it fills the tube up to the water level. A mantle of ice soon forms around the tube, and after 10 to 15 minutes, it is fully formed. The remaining carbon dioxide can be tipped out. Notice that the ice mantle looks bigger than it really is. This can be seen when the cell is inverted. It should never be allowed to grow to the outer wall of the cell, and especially not to bridge across the surface for fear of fracturing the glass. An alternative method of preparation is to use brass rods cooled in liquid nitrogen. In this case, the thermometer well should contain an organic solvent such as acetone, almost up to the water level. A cold rod is gently inserted into the well. The initial bubbling seen here is caused by the evaporation of drops of liquid nitrogen transferred with the rods. The rod is left in place for about half a minute before being replaced with another. 15 or 20 insertions may be needed to produce enough ice in the cell. When the ice sheath has fully formed, the acetone is poured away. It is replaced by ice-cold water, or, if necessary, an electrically insulating liquid. This ensures good thermal contact between the thermometer and the contents of the cell. After preparation, the cell should be returned to the ice for some hours to come to equilibrium, and it should not subsequently be inverted. In making the cell ready for measurements, it's necessary to free the ice mantle from the central tube. This is done by briefly inserting an uncooled rod to melt a thin layer of the ice all around the tube. One can verify that the mantle is free by giving the cell a short axial rotation and observing that the mantle is not attached to it. This is crucial because it is the ice-water interface around the thermometer well which ensures that the triple point condition prevails at the center. A thermometer inserted in the cell will then take up the correct temperature. The cell is kept and used in ice. The thermometer to be calibrated is first cooled in the ice and then transferred from the ice to the cell. The most accurate thermometers are platinum resistance thermometers whose electrical resistance varies strongly with temperature. To make the reading, the thermometer is connected to a sensitive resistance bridge which is balanced so that the meter reads 0. The resistance can then be read from the dials, the last place here corresponding to 10 microohms or 1/10,000th of a degree. The stability of the reading can be seen on the chart recording. The step change being 1/1000th of a degree.
We had a problem, that of demonstrating that we had a thermal gradient that was not greater than plus or minus 1 degree C at approximately 120 degrees C.
Root's problem arises in the autoclaves used to sterilize surgical products. Each day, thousands of samples must receive a prescribed heat treatment. DHSS regulations for these products specify very tight limits of measurement and control of the sterilization temperature.
It is surprisingly difficult to obtain 9 commercially available thermosensors which, when physically connected together and subject to the ice point and then the steam point, will agree with each other. And if we can get them to agree within 0.3 degrees C of each other, we're doing pretty well. What we really wanted was a checkpoint at the working temperature. About a couple of years ago, I went to an instrument exhibition and visited the NPL stand, where they had some triple cells on view. I was familiar with the water triple cells, but they also had a couple of organic cells at 26 and 36 degrees approximately.